Hello everyone, myself Manvi Devatia, student of MSc Biotechnology Honors. Today we are discussing about on topic Field Emission Scanning Electron Microscope, which is also known as FESEM. So there is an overview. So we are discussing about it introduction, principle, instrumentation and working, application, advantages and its conclusion. So there is an introduction of FESEM. So FESEM stands for Field Emission Scanning Electron Microscope, and FESEM is a microscope that works with electron particles with negative charges. These electrons are liberated by a field emission sources, and these objects scanned by electron according to a zigzag pattern also. Its high resolution and depth of field allow for detailed examinations of cellular and subcellular structures. It also detects morphological changes associated with diseases and a nanomaterial based diagnostic tools. Further, we are discussing about its principles. So there, as we show in this diagram, there is gun with filament, multi-hole apertures, linear tubes, columns, chambers, wells, and many more things. So FESM used a focused beam of electron to generate an image or analyze the specimen. For operation, the gun head and the column and the specimen chamber has to be evacuated. Vacuum in the specimen chamber is measured by panning gauge, and column chamber value remains closed until the detection pressure is not ready for operation. After vent command, column chamber value closes and N2 gases flows into the specimen chamber through vent value, and a field emission cathode in an electron gun of a scanning electron microscope provides a narrow probing beam to low as well as a high electron energies. Further, we are going towards instrumentation and working. So as the diagram shows, there is electron beam coming and there is anode to detect it, and there is magnetic lens also, and then it goes to a scanning coil which scans, and after scanning there is backscattered electron detectors, and it shows to its scanner or the TV, which is a computer which is attached to it. So the field emission scanning electron is an instrument which, just like the SEM, provide a wide variety of information from the sample surface but with a higher resolutions and a much greater energy range. It works just like a conventional SEM. The sample surface is scanned with an electron beam, while a monitor displays the information that interests us on the basis of a detector available. The biggest difference between a FESEM and SEM lies in the electron generation system. Another highly remarkable feature of FESEM its use in lens detector. These detectors, which are optimized to work at highest resolutions and very low acceleration potentials, are fundamental for getting the maximum performance from the equipment. And FESEM uses a field emission gun that provide extremely focused high and low energy electron beam also. Further, we are going towards its application. So there are four images: SEM image, TEM image, AFM image, and FESEM image. So there are applications. Its application shows a measurement of samples microscopic features. It tells about the study of surface morphology, characterization of coating, integrated circuit evaluation, fine structure analysis, study of microstructures, fractures, and coating thickness measurement and cross-section analysis. It also tells about the 3D imaging. An FESM is used to visualize very small topographic details on the surface of entire or fractional object. It also tells that the FESM may be employed for study organelles and DNA material in cells, synthetic polymers, and coating and microchips also. So there are some advantages. The ability to scan smaller area contamination spots at electron accelerating voltage compatible with energy dispersive spectroscopy, high quality low voltage images with negligible electrical charging of sample accelerating voltage ranging from 0.5 kV to 30 kV. Essentially, to no need for placing conducting coating on insulating materials, and observation of very fine surface features of coating material and pharmaceutical dosage form. And lastly, there is in conclusion. So in conclusion, FESEM is a powerful imagining techniques that provide extremely high resolution and high magnification images of sample surface. Utilized a field emission electron sources, FESM offers superior image clarity, great depth of field, and minimal sample damage compared to conventional SEM. It is widely used in material science, biological, nanotechnology, and semiconductor industries also. Overall, FESM is an essential tool for advancing research, development in various scientific and industrial fields due to its precision and versatility. Thank you.